Okay, well, let's get started. So today, the plan is that we need to finish Chapter 2 homework discussion. We have like 2 more questions to go through. And then after that, we're going to start into Chapter 3. And those slides went up sometime over the past several days. I can't remember when. Maybe Sunday night. I have been meaning to tell you, and I keep forgetting, so I put it in blue on the announcement thing. You are going to need a calculator that has a log button on it. In the past, I have let people use their phones as a calculator with a log button, and that's fine. The rule is you can only use your calculator on your phone during quizzes and exams for that particular purpose. And you're going to have to learn how to figure out how to use the log button, which varies from calculator to calculator depending on how you input the numbers. But I'll be happy to work with you on that. And we will have some practice both on the homework for Chapter 3 as well as just right in the middle of lecture for it. On Friday, I would also like to do a quiz, and the quiz would be on Chapter 2. So the quiz would be it's a short quiz. It would only be 10 points. This is not intended to be a big deal, but it is kind of a checkup for you, so you get a chance to assess where you are in terms of your understanding of some of the key concepts and facts and stuff. So the quiz would be based on lecture notes from Chapter 2 as well as the homework discussion from Chapter 2. If there's stuff that's in the textbook that we didn't talk about in lecture or on the homework, then it's not going to appear on the quiz. Problem. I'm not going to intentionally do that anyway. And then once we finish the quiz, we'll continue on into Chapter 3. Let's see. I think that's the major stuff. Well, and then Monday, there will be a homework for Chapter 3. The homework for Chapter 3 will be a little bit different from probably all the other chapters in that this chapter requires a lot of, I think a big part of it is learning how to do some calculations as well as some more qualitative sketching and stuff. So it's going to be done in hard copy. It's not going to be submitted electronically. I'm going to give you the sheet. I don't know. It's online, but I'll probably give you the sheet that you'll complete on Friday, and then it'll be due on Monday. And my suggestion is I'm going to collect them at the start of class, but then I want to go over them. So I would suggest that you bring a photocopy. That way you have the thing in hand and you'll be able to take notes directly on it. Also, I am going to be grading it because it is problems. So I will actually be grading that one for accuracy. But you'll learn how to do all this stuff during the actual lecture. I think that's my comments for now. Any questions? So if not, let's switch back over to the homework. So we were, you have to remind me, where was the last group to talk? And I can't remember. I kind of feel like you were back there, right? So I think that side hasn't talked yet. So we have 2 more questions. So maybe we can break these into a few. How many groups are over here? Is it 2 groups or is it 3? Like there's one in the back, right? And then is there another one sort of middle row and then another one here? Something like that. Okay, so we'll break it into 3 so everybody gets a chance to chime in. So if we could get the back row back there, if you could sort of chime in on question 5 and just describe your answers for this one. So what does horseradish peroxidase do for us?
It is taken up by neuron axons, packaged and vascular transported by diagonally through retrograde transport to the soma of the cell. And this causes the entire neurons to become radioactive, so you can trace the path that has been made between neurons. And it can be useful in a neuroscience laboratory because you can trace connections from the terminal to the source.
Right. So did you guys have like a research idea or like topic or something like that? Probably each person's different, but what did you put down?
Said like how you could trace how like the rabies virus reaches the brain that's been injected.
Yeah, because it works in a similar way, right? It's retrograde transported. Yeah, so maybe you're effectively following the rabies virus through its pathway. Did anybody else have anything different for A? Or any additional stuff for A? Or that's pretty much, I think, the answer. I mean, that you're basically dealing. The cool thing about this particular stain is that it's transported into the neurons that light up in terms of where the cell bodies are that light up. That stain gets there by retrograde transport going back up axons. So you can use this to kind of reverse engineer neural pathways, which I think is the neat thing about it. What about group B here? Group B. Middle group, the middle row here. Did you guys have a different kind of a study for part B here that you put down?
We said maybe like trace motor pathways in the brain.
Yeah, so if you know where I guess ultimately if you're dealing with like skeletal motor pathways, if you were to inject around the neuromuscular junction, you could actually trace that back up and find out where in the spinal cord the cell bodies are those neurons that were innervating that skeletal muscle. And then once you know that, you could inject at that site and then trace further on up the spinal cord or into the medulla and keep tracing on up. And through that way, you could kind of reconstruct a motor pathway. I think that would work. Does anybody else have anything cool they thought of that you could do with this? Yeah, what'd you think?
I don't know if this counts.
Okay.
But I said which neurons initiate the HPA axis.
What's the HPA axis?
Hypothalamus, adrenal, pituitary.
Yeah, so if you're looking at that axis, I mean, it sort of depends. So, okay, so if you're looking at hypothalamus releasing hormones going to the pituitary and then going to the adrenal gland, for example, I don't think this would work because you're not tracing neurons then. But if you look in the posterior pituitary, and you might know this, other folks, if you've taken a physiology course, for example, the posterior pituitary contains axons that are coming from the hypothalamus, and they're releasing hormones into the blood from the posterior pituitary. So you could inject the posterior pituitary, and this stuff would presumably get taken up by those axons and travel back up the hypothalamus, and you can see where those cell bodies are originating. I think that works. I'm maybe going to say this has a proper name, but without getting into too much detail. So that works. I mean, I think you could do, this is the idea though, is that you're basically reverse engineering different kinds of traces around the brain. So if I was interested in, what was my example? We talked about Broca's area in chapter 1, which is this language motor area. And so if I was interested in what are the inputs that are sent to that motor area that controls the language parts of your mouth, I could inject horseradish peroxidase into that area, the Broca's area, and that stain would then effectively travel to all the inputs that go into Broca's area. So you would expect it to go from, there's another area called Wernicke's area, for example, that informs Broca's area about what to do. You would expect to be able to trace back to Wernicke's area using this horseradish peroxidase, and probably other areas that are also coming in. So there's, this is, I think, the utility of that particular stain. Make sense? Questions? So certainly, I mean, we have 3 total stains we've talked about. We're not going into tremendous detail on methodologies, but we've got horseradish peroxidase staining. We've got the Nissl stain, which is just the sort of generic, basic, purple, pink type of stainings. And then we've got the Golgi stain, which allows us to visualize for at least a subset of the neurons in your tissue slice. It allows you to visualize the entire neuron from cell body out to the tips of the dendrites down to the bits at the end of the axon. And you should be comfortable knowing what each one does and how you might use them differently. Okay, number 6. So I'm going to go back to the middle row there. Can you start us off? So what I'm going to ask you to do is maybe give 2 of the different glial cells, then we'll go to the front row and we'll do the last 3. I wrote them up here just for clarity, but you don't have to go in that order. So whatever order you have it in, you can just give 2 of them.
Okay, so the astrocytes regulate the chemical content of the extracellular space, and then the oligodendrocytes myelinate neurons in the brain and spinal cord.
Okay. front row. Is your speaker here? Okay.
The Schwann cells are found in the peripheral nervous system, and they provide layers of insulation to the neuron membranes, so they do myelination. The epidermal cells are in the central nervous system. They are fluid-lined ventricles in the brain that direct cell migration and cell development.
Let me put this. Get rid of that one. Microglia. Did you get that?
They're phagocytes in the nervous system, and they remove debris from dead or dying neurons.
Put and other garbage too. Anybody else put anything additional for any of these cells? I think this is sort of the core of it. I mean, these guys do, I mean, these are the 5 principal kinds. There's another kind you'll see in some textbooks called satellite cells that do a lot of the same kind of regulatory stuff that astrocytes do, except they're doing it in the peripheral nervous system. So astrocytes are primarily a central nervous system cell, as are then the oligodendrobilia. The only peripheral nervous system cell actually in this list are the Schwann cells. These guys are CNS, these guys are CNS as far as I know. The only one I would put anything additional on really is the astrocytes, in that they do a lot of stuff. So they definitely are heavily involved in regulating the chemical content of the extracellular space, and we'll even talk about that a bit in chapter 3. How one of the critical roles is they help to move potassium away from neurons. So they bring in potassium and then they route it away from neurons, because potassium can have a sort of volatilizing effect on neighboring cell membranes, especially of those neurons that are near them. But they also do some other things. So you might have heard of, probably heard of the blood-brain barrier, which is basically this sort of continuous barrier that separates the contents of the blood from whatever is in the brain, and really, frankly, whatever is within the central nervous system. And the way that basically works is any of the capillaries that are routing through the brain are basically just completely surrounded and enclosed by astrocytes. And so the astrocytes also function then as a blood-brain barrier, as the blood-brain barrier, in that, or at least they're responsible for it, because they basically form an additional layer of protection around the blood, all the blood vessels, including all the capillaries, that prevents material, by and large, from getting in and out of the brain from the blood. So there's sort of an extra hurdle to get anything from the blood to the brain. Some stuff certainly passes through, like glucose passes through readily, gases pass through regularly, other nutrients pass through readily, and there are specific transporters that are on those astrocytes that allow that to happen in many cases. But things like, drugs or bacteria or poisons or whatnot actually have a very difficult time penetrating that barrier, and the reason is largely due to these astrocytes. There's other cool things about astrocytes. There are some, like I've seen some papers in the last 5 years or so that have indicated that astrocytes actually can play a role in modulating how neurons respond to other neurons. So this means that they are reacting to the inputs that neurons are receiving, and they're helping to regulate the activities of neurons. So that means on some level they might actually be involved in some decision-making processes, like cellular level decision-making about when to fire, when not to fire. They seem to be really interesting, is sort of the take home. They do a lot of really cool things, and I think we're going to increasingly see and learn more and more about the importance of these astrocytes as over the next 10 years or so. Let's see. The other thing I'll just mention is that many of you probably are aware of this, but the myelination that the oligodendroglial do and the Schwann cells do, the main thing that does is that it helps to speed up the rate at which signals move down the axons. So they're insulating them, and what that insulation does is basically it prevents signal loss. And by preventing signal loss, you're able to upregulate the degree to which materials can move down those axons. regulate the rate at which they move, the signals move down axons. So, and it's, you know, it's hundreds-fold types of increases. We're going from 2 meters per second without myelin to 150, 200 meters per second. So we're doing a massive increase, several orders of magnitude in terms of increase in conduction rates. So that, you know, glial cells are really important, and we will mention them now and then. We certainly will talk about the myelinating glial cells, the oligodendroglial and the Schwann cells. These other guys we probably won't spend that much time on this semester, and I kind of regret that. They're definitely getting short shrift, but I think it's, you know, we're going to have to focus on neurons mostly. But one needs to be aware that a lot of the pathologies that you guys might end up investigating for your term papers end up involving some of these glial cells in many cases. And so there's a good chance you're going to encounter these things again. Any questions about anything related to part of the homework? So again, we'll do a quiz on Friday. Quizzes are not meant to be a big anxiety triggering event. They're just meant to be a little checkup. They're not worth a ton of points. No one quiz is going to hurt you or really kill you, even hurt you that bad in terms of final grade. But it is an opportunity to sort of induce some studying and also, you know, encourage you to get a feel for where you are in the course. Yes, sir. It varies. My goal when I start writing a quiz is usually to write it as a multiple choice quiz, but then inspiration strikes. So sometimes they will end up being pure multiple choice. Sometimes they will be some mixture. Sometimes they will be short answer. Sometimes there's matching. Sometimes there's some little sketch. Honestly, I'm sorry, I just don't know until I write the thing, and I haven't started thinking about writing this thing yet. So I don't know. One thing I will say is that you're not going to have to fill in blank without a word bank. So you need to know your terms, but you probably don't need to know how to spell them yet. On the exams, there will be a fill in the blank section that doesn't have a word bank. But on these, if there is fill in the blank, there will be some manner of word bank or something. Other questions on that? I think it'll take like 10 minutes, hopefully at the start of class, and that probably is it. That's really usually the intent. And we'll go over it right after we get done taking it. I'd like to launch into Chapter 3 now. And so here we're kind of stepping, in many cases, we'll be stepping past some things that you've seen in other classes. If you've had a lot of basic cell biology before, some of this stuff is likely to look a little new. I mean, if you've had physiology courses, you've heard of some of these things, but I bet some things will be more novel concepts for you in this chapter. So the way this chapter is devoted, this chapter is titled the Cell Membrane at Rest. Right? So we're interested in what's going on within a resting neuron in this chapter, and then the next chapter we're going to actually stimulate that neuron and go through everything that happens during the stimulation, and particularly what happens during an action potential that's traveling down an axon. So to understand how membranes work, we're going to spend this chapter and first introducing us to membranes and what are called membrane potentials, and we'll talk about the chemical players that are involved as well as what drives them, chemical and concentration gradients. And we'll talk about and use some of the equations that are used to actually predict quantitatively exactly what the membrane potential should be for a given set of conditions based on the relative concentrations of the ions and the relative permeabilities of those ions. Then we'll talk a bit about potassium structure and selectivity. And then the homework, which is due Monday, is going to involve a lot of practice with some of the calculations using these equations that we're going to be doing, as well as what the role of potassium in astrocytes and lethal injections. So it's a fun topic. Okay, so if you think about what neurons have to do, the real challenge they have is that they are using electrical signals and they're sending electrical signals along cells, but they're doing so in water. And any time you may have in the past tried to take an electrical appliance and submerge it in water and then plug it in, it usually doesn't work very well in water. And that's because water tends to grab up spare electrons really readily, and it disrupts any kind of electrical signals that you might be trying to transmit. So if you sort of think of axons basically as wires, and you sort of draw the analog to telephone wires, like axons, have to conduct electricity over a distance, but they have a lot of advantages over what neurons can do. Because in a telephone wire, you have this really superb conductor. This is like a traditional telephone wire. You have a nice copper wire running down the inside of there, and it's surrounded by sort of a rubberized insulator. So you have this beautiful conductor, and it's surrounded by insulation so that there's no other interactions with that conductor. And electrons can just zip from copper atom to copper atom all the way down that wire in order to send a signal. In contrast with electrons, you can't really do that because for a couple reasons. One is you're not just dealing with electrons, and you're not just conducting electrons. The electrical signals in axons are actually ions. They are sodium or potassium or calcium ions, mostly sodium is what we'll be focusing on, that have to travel through this aqueous medium. So that is sort of a problem. Each of those ions will interact very readily with water molecules. They're very strongly attracted to water molecules, and therefore water can potentially impose a pretty substantial penalty on their movement. And so as a result, they're very prone to signal loss because of this interaction of ions with water, as well as the potential for ions to leak across membranes, which sort of causes you to lose your signals. So the solution for this, the way axons are actually able to function, is that they're not just sort of like a passive electron conductor like a copper wire is. Instead, these are very active membranes. So for an axon to transmit its electrical signal, it is actually generating a signal each incremental step down the membrane of the axon. It has to regenerate and amplify that particular signal. And we're going to be talking about that in this and the next chapter. We say that they have excitable membranes because they're able to, and it's not just one part of the membrane, it's the entire stretch of membrane along the axon. So again, this chapter we're going to be studying membranes at rest, and then in the next chapter we're going to be looking at how the membrane gets excited to carry information, and how that process works. And then ultimately in the chapter 5, which is sort of the last chapter in this unit, we'll be getting to the end of the axon. The action potential would have traveled all the way up the axon at that point, and we'll be seeing how neurons communicate from one cell to the next, and how those signals are received and interpreted, and ultimately can produce another action potential in the next cell down the line. So we're going to try to go through that whole process over these next several chapters. Okay, so in terms of setting this up, the basic unit that we'll be looking at that's responsible for these electrical signals are ions. So ions, if you've had any chemistry at all, these are charged particles. They strongly attract water molecules, and therefore they dissolve readily. I mean, and here we're talking about things, mostly we're talking about sodium ions and potassium ions, but other ions come into play as well. We'll talk about chloride ions a bit, we'll talk about calcium ions a bit. And all of these things are charged particles that interact very well with water. There are two main kinds, just as sort of a recap from high school. So cations are positively charged ions, and anions are negatively charged ions. I had a physics professor in high school that really liked cats, and so I put this on the front page here. So this is not his thing. I just saw this over the summer, and I was so happy because Mr. Radloff kind of started this in my mind. So my high school physics teacher, he felt that cats were very positive creatures and had positive charges, and he even did this terrible pun, that they have a positive charge because they're cats and positive creatures. And so as a result, we call cations. They're positively charged particles. So that would be sodium and potassium primarily that we're going to be dealing with. Whereas anions are all the negatively charged ones. As I said, cations and anions will interact very strongly with water molecules. And the reason is that water, while not itself charged, is a highly polar molecule. So when you look at water, we all know water is H2O. So that means it's got two hydrogen atoms and an oxygen atom. The oxygen atom has very strong electronegativity, which means it's highly attracted, and electrons are highly attracted to oxygen molecules. Whereas hydrogen has much lower electronegativity and therefore is not as attracted to the electrons. This is a covalently bonded molecule, which means that electrons are freely flowing from one atom to the next, and they're circling all around them, and you can know either how does it work that you can know how fast they're going or where they are, but not both, because that's what physics does at very low scales. But the key thing is that because they are more strongly attracted to oxygen, electrons are more likely to be around the oxygen atom than they are to be around the hydrogen atoms at any given moment. And therefore, the oxygen side of the water molecule has a partial negative charge, and the hydrogen side of the molecule has a partial positive charge. And that positive charge, while it doesn't result in the whole molecule actually being charged, it does mean that if you've got a positively charged ion, like these sodium ions, which are shown in red here, the sodium ions will be very attracted to the negatively charged part of the water molecule. And so you get the oxygens basically clumping around the sodium molecules whenever they go off into solution. And similarly, here is a chloride, this would be an anion. It would be negatively charged, so it would be attracted to the hydrogen part of the water molecules. And you can see that's the part of the water molecule that's gliding around them. So that means that water interacts very strongly with all these ions. It also means that that these ions dissolve readily in water, and that's really what's happening here. You get this is called like a hydration shell that's forming around each of these ions as that ion gets dissolved into solution. So we're interested in ions, ultimately. And the thing that ions do then that is so interesting to us in terms of being able to drive or cause electrical signals on a cellular membrane is they have to cross membranes. And that's kind of an interesting problem. So here's a biological membrane, and we talked about this in the last chapter. All neurons, like all cells, are composed of, they're surrounded by a phospholipid bilayer that is their cell membrane. The phospholipid bilayer has both phospholipids and proteins, and I like this picture quite a bit. I ripped this off the internet sometime in the past, and I don't know where I got it from, but it's nice because it sort of shows, it's basically a ball of a cell that has sort of just a notch that's been cut out of it, and you can see how the phospholipid membrane is a part of, or composes that cell membrane. So how you have this little phospholipid bilayer, these two layers of phospholipids. And as I mentioned, you've got two groups on a phospholipid. You have the polar head groups, which always orient towards the water. So they orient towards the outside of the cell as well as the inside of the cell. And then you have this huge band of hydrophobic fatty acids that's along the inside. That band of hydrophobic fatty acids then does not interact strongly with water, and it gets marginalized and excluded and pushed basically into the center. And as I mentioned, if you take phospholipids and toss them in water and shake them up and look under the microscope, you will actually find spontaneously formed cell-like structures like this just because of the hydrophobic and hydrophilic nature of those molecules. So it's, they're really cool molecules in and of themselves. But because you get this hydrophobic layer, that means that charged things that are strongly attracted to water really can't penetrate through the cell membrane. Quite literally what happens, if you sort of imagine, let me go back a slide. If you imagine a little sodium ion that's really strongly attracted to all these water molecules, and so it's got hydrophobic, it's got hydrogen forces basically that are causing these attractions. It moves into an area over here where it can interact readily and also be attracted to these hydrophilic head groups. But if the sodium ion starts to go into this area in here, the hydrophobic zone, it's all of a sudden going from an area where there's tons of water and it's all attracted to everything to this area where it's not attracted to anything. And it literally just gets pulled back out. And so as a result, ions just don't cross through this hydrophobic layer. They get pulled back out by all the water and other hydrophilic things that are outside the membrane, and they just can't go through, at least not at any kind of a reasonably decent rate. There's nothing really magical about it. It's just, they're really attracted to all the stuff that's outside or inside the cell, and they're not attracted to anything in here, and so they don't readily cross. That's kind of how I think about it, and I think that makes a lot of sense on a mechanistic level. So how do ions get across? Because I said we've got to get ions to pass across membranes in order to cause some of these electrical disturbances, like membrane potentials that we're going to be tracking. And that's where the other component of membranes comes in. And that other component are the proteins. Any questions on this stuff, though? Like phospholipids? I feel like we don't always explain why things are that much sometimes in like intro bio. We just sort of say, ions can't get across the membrane. And we don't really explain why. So that's why, in a simplified sense. So how do you get across? The key thing is you have to have proteins then that are available to allow hydrophilic compounds, which would be charged or polar compounds, to get across the membrane. And those, the proteins then can be channels, or they can be pumps, or receptors, or enzymes. There's all kinds of things proteins can do. But in all cases, proteins are just long amino acid chains that were formed by that whole process we went through in the last chapter with transcription to make RNA, and then translation to turn that into protein, which then has to undergo post-translational modification to form a nice mature protein. So here you've got a nice chain of amino acids that they're depicting that then gets folded into discrete structures. So here it's folding into an alpha helix, and then multiple alpha helices get combined to form this subunit. And in this case, you get multiple subunits combining, and now you're forming this tube-like protein complex where, maybe you can't totally tell, but it's basically hollow, where you've got these subunits around the outside that form kind of the walls of a tube or a channel through which now maybe a particular ion can pass through. This is a really common arrangement. This is how most ions get across membranes, is via channels like this. They're passive, so they're just basically pores. They could sometimes be opened and closed. And when they are gated like that, when they can be opened and closed, we call it gated. There's two main ways they'll be gated. One way is called a voltage-gated channel. The other one's called a ligand-gated channel. And the idea is that a voltage-gated channel will open and close depending on the charge environment around it. So as you start to become more positive or more negative, the thing will open in response to that change in the charge environment around it. Whereas a ligand-gated channel would be a channel that opens or closes in response to something else binding to it. So a good example of this is that when a neurotransmitter comes across and binds to receptor, sometimes that receptor also has a channel in it. So you bind the neurotransmitter is the ligand, and at that point then the gate opens. That's what we call a ligand-gated channel, because it's opening in response to binding a neurotransmitter. Many of these channels are really, really specific. So they will quite literally sometimes allow only one kind of ion to get through them. And that's usually dependent on the very precise size of the channels as well as the charge environment that aligns them. And we'll be talking about potassium channels in this chapter and a little bit about how it is that they are so specific to just allowing potassium ions, potassium is the K, to just allowing potassium ions to go through them. Okay, so we've got the major players now. We've got ions, we've got water, we've got membranes, we've got channels in the membranes. So the key question then is that if you're looking at a membrane, marker, so here's a cell, and let's even say that you've got a channel. This is how I draw channels. I don't know, this is what I've always done. So that's supposed to be, that's like a cut through the channel, so the whole thing would be like a ring-like structure, and that's the hollow bit inside the channel. That's how I've always drawn them. So let's say you've got a cell, that's a cell, you've got a membrane, you've got a channel. This channel could allow an ion to go through it, so maybe it's a potassium ion. The question is, what might drive ions to move through a channel in a really directional way? Why would you ever get a situation where you're having large numbers of ions moving from one side of a membrane to another? So, you could imagine if you've got potassium could go this way, it could just as easily go this way. But if you're going to try to get directional movement, so if you're trying to get potassium ions to go from the inside to the outside, or the outside to the inside, when would that happen? Like, what would be the circumstances that would cause that? What do you think?
Concentration gradient.
Okay, so maybe you have a concentration gradient difference. So if there's large numbers of potassium ions out here, I'm drawing it as a big potassium, but that means large numbers of potassium ions and very few potassium ions inside, then you might expect, we sort of learned this by rote, things will tend to move from high concentration to low concentration, so you would expect to see a net movement of potassium ions from the outside of the membrane to the inside of the membrane in that circumstance. Make sense? So this is one way by which we can have movement just by simple diffusion. So this is a diffusion-based motion where you get a high concentration on one side and a low concentration on the other. We call that a concentration gradient, and that term's on the slide there. And that can cause movement by diffusion. Again, I kind of think that sometimes we do a good job of telling people how things work and what things do without necessarily explaining how they work. Let me pull up a video here. So the key thing about diffusion, I think, that's interesting is that it's not, that diffusion is just random motion of molecules. Just waiting for the video to show up here. So we're watching molecules move around randomly, and that somehow is causing this net movement of particles. And I think this video shows this pretty well. So in this animation, what we're doing is we're starting with, you can sort of think of this as a membrane here. This would be maybe inside the cell, this would be outside the cell. But the key thing is that you've got a high concentration over here, you have a membrane, and then you've got a low concentration over here. And if you look at any one molecule as we start this animation, each molecule is moving in a random direction. It's not like all the molecules are moving this way because they can somehow tell if they're at high concentration. What happens is they're all moving around randomly. But because there's lots of them here and there's quite literally none over here to start with, it's much more likely that some of the ions are going to go this way and very unlikely that any ions are going to go this way. So you get this net movement from the high concentration to the low concentration as a result of just random motion of each individual particle. So let me hit the play button here. So initially, again, we've got high concentration down here below and low concentration up here. But because there's many, many more particles down here that are all moving randomly, it's much more likely that particles will move from the lower part of the animation up into the upper part of the animation. And it's very unlikely, until we get to the later stages of the animation, that you get particles that are going from up here down back into the lower area, just because there's so few of them. So you're just relying on a random chance thing, but there's very few out here that could randomly go down. There's many more down here that could randomly just end up going up. So each particle is moving randomly. What we're interested in is what's the net movement of all those particles. And so that's basically looking at the number that are going from the high concentration to low concentration, minus the ones that are going from the high to the low concentration to the high. So we're interested in that net movement whenever we're talking about diffusion across a membrane. Does that make sense, more or less? I think that's kind of a critical concept. Okay, so these ion channels are going to allow us to have some kind of directional movement that occurs across membranes, just simply by having an opening that would allow ions to move from one side to the other if there's some kind of a concentration gradient. So if here's a scenario, this is from your book. Here we're starting with a scenario where there's high concentration over here and low concentration over here. And in this case, initially, this is just a membrane with absolutely no pores in it, no channels. So if there's no channels, we can't get any movement across the membrane. So I have a little prompt here. Why is that, do you think? Why can't we get from the left side to the right side if there's no channels? Yeah. You can't go through the membrane, right? So it's just kind of redundant here. But in this case, we've got absolutely no permeability. There's no channels, and therefore ions can't move. So that's just sort of basic, help us remember how membranes work. If, however, we open some channels, in this case we've opened a sodium channel and a chloride channel, now the ions can move from the left side over here over to the right side by going through their particular channel. Okay? And they're going to tend to move in a directional fashion, where we'll have net movement by diffusion from high concentration to low concentration. And that would continue until such time that we got equal concentrations on both sides. And at that point, there's no net movement across the membrane anymore. There actually still is movement, so we still would have ions that sometimes would go from this side to this side randomly, and sometimes ions would go from this side to this side, but they're doing so at equal rates now because the concentration is the same. There's equal numbers on each side, so there's an equal probability that one can cross from the left to the right as there is from crossing from the right to the left. And so at this point, we would have exhausted our concentration gradient, and while there's still occasional ions going back and forth, there's no net movement. We're not going in a net sense. We're not seeing an increase or decrease of ions on one side or the other. All right, so that's basic diffusion across a membrane. Here's a wrinkle to it, though. Well, now before I get to that, I should say, so if this is one way that we can get ions to flow across the membrane, the next question is, where do these concentration gradients come from at all? And you probably know the answer if you think about it for a second. Most of the time, we get these concentration gradients established due to the action of ion pumps that are pumping ions across membranes. So the most abundant, most common, most important pump of any sort that we have is something called the sodium-potassium pump. It's also referred to as the sodium-potassium ATPase. The ATPase thing indicates that it's an enzyme that breaks down ATP, and it uses the energy from ATP, that came from mitochondria probably, to power the pumping of sodium ions and potassium ions. These pumps work by pumping potassium ions in, and they pump sodium ions out. And technically, they move 3 sodium out and 2 potassium in for each cycle that they complete. I tend to not try to dwell too much on the fact that there's 3 versus 2. There's other factors that kind of ends up balancing that thing out. The key thing is that sodium is almost always pumped out, and potassium is almost always pumped in. And so as a result, we tend to have a concentration gradient in most cells such that there's high concentration of potassium ions inside cells. It's actually the opposite of what we drew. There tends to be a high concentration, because we've got these pumps, there tends to be a high concentration of potassium inside cells and a high concentration of sodium outside of cells, pretty much universally. So this is, if you compare intracellular fluid concentrations of these ions, you tend to see lots of potassium inside and lots of sodium outside. And that's probably the single biggest difference in ions that you see between the inside and outside of most cells. There are tons of these pumps all over. Many, if not most, of our cells have these. They require a tremendous amount of energy. In our brain, certainly all of our neurons have tons and tons and tons of these things, and they are responsible for something like 70% of our brain's ATP use is just used to power these pumps. So it's a really critical thing. If you're using 70% of your brain's energy, and we said the brain is like 10 times as metabolically active as most other tissues, and 70% of that activity, 70% of that metabolism is used to pump sodium and potassium, that certainly indicates to you that these ion concentration gradients, having high potassium inside and then high sodium outside, those concentration gradients must be really important in terms of driving the function and activities of neurons. There's other kinds of pumps that we'll see as well, so it's not just sodium, potassium, ATPase, that's the most important, but we will talk a little bit about calcium pumps, for example. We have calcium pumps that can pump calcium usually out of a cell or sequester it into the endoplasmic reticulum. That's really important in muscle cells, actually. That's how they are able to sequester all their calcium within the sarcoplasmic reticulum. But neurons do the same thing. There's also things called proton pumps, which we're not going to get into too much, but the mechanism by which mitochondria are able to create ATP relies on using proton pumps to pump protons, which are little hydrogen ions, out of mitochondria and create it's called a proton motive force, but it's basically just a concentration gradient of hydrogen ions across the membranes of the mitochondria. That's part of the electron transport chain. Here's finally, I forgot to put a picture up. So again, here's a picture of the sodium-potassium ATPase, sodium-potassium pump. So in this case, it's taking sodium from inside the cell, and it's taking potassium from outside the cell, and basically it just sort of switches conformations. And each cycle, it'll start with being open to the inside of the cell, at which point it'll grab up sodium and release potassium, and then it changes conformation such that now it's open to the outside of the cell. At this point, the sodium that it grabbed before gets released out of the cell, and potassium ions get grabbed, and then it cycles again and pulled back in. And each cycle, it burns ATP. So that's the ATP being consumed within each cycle that allows that process to be powered. Yeah?
You keep saying that it grabs the ions from outside of the cell. Does it actually draw them towards it, or do they just randomly fall into it like they do the concentration gradient?
It's more of a grab, I tend to think of it. So each of these little slots has a particular size and charge that is just right to fit a sodium ion. These would be just right to fit a potassium ion. So they are effectively grabbed. They're attracted into those places. And then when the conformation shifts, the size and structure of those changes a little bit. I don't exactly know details on exactly what happens, but I think basically the size changes a little bit such that now sodium ions are no longer as attracted and they just sort of pop right out. So it really is more of like a grab using electrical forces. They're attracted to those slots. Okay? Yeah?
So now, are those pumps able to change which, if it's like if it's made for sodium, is it able to change into like a potassium? Like it's just kind of set in stone?
Those are really set in stone, yeah. The only thing they can do is pump sodium out and potassium in.
Okay, so it's not like it can change depending on any.
Not that I'm aware of. I mean, over evolutionary time, you could have changes in the protein, but within an individual, this is something that's basically driven by the particular genes they've got, and that's what those proteins do. Questions? Anything else? Okay. So this is indicating how ions can be driven across the membrane by a concentration gradient. It's explaining where the concentration gradients come from, which is what this slide's about. They come from these pumps. So the pump sets up the concentration gradient, and then you have ions that, once you've got sodium at high concentration out here, if there's a sodium-specific channel, now the sodium ions at high concentration can diffuse passively back through that channel. So we're kind of setting up these loops to happen. What we haven't explained is what this has to do with any kind of electrical disturbance, right? I mean, at this point, there really hasn't been any kind of electrical disturbance. We don't have any charge differences per se across the membrane. We've got positive things coming out and positive things coming in, but it's neutral. I know it's 3 sodium going out and 2 going in, but again, think of the pump as a neutral structure. So given that, where are these membrane disturbances coming from that I keep mentioning? Where are we getting these membrane potentials? And to understand that, I think we have to go back to that same thing we had on the prior slide. So here we've got a positively charged ion, in this case it's potassium, and negatively charged ions, in this case they're just calling it an anion. I think before we might have called it chloride on the prior slide. In this case, now what can happen is we could open a channel that only allows potassium to go through, but that same channel might not allow the anion to go through. So if the anion can't get through the membrane, now we've got a kind of a weird situation, right? So here we're starting from scratch. This is just a membrane. There's no channels at this point. And if there's no channels, then there's no net movement of this particle across here, despite the fact that we've got concentration gradients for both the cation, the potassium ion, and the anion. If we just open a potassium channel, though, and we don't open an anion channel, now the potassium ions are free to start flowing down their concentration gradient, and they will. We'll start to get randomly some potassium ions that will make it through the channel. But the anion is stuck over here because it can't go through that same channel. So if this guy's stuck and potassium ions are flowing across, now all of a sudden we have this net movement of positively charged ions across the membrane that is not balanced by a movement of negatively charged ions. These guys are stuck over here at high concentration. And so because we have this movement of these positively charged ions to this side of the membrane, now all of a sudden, right along the inside of the membrane, these potassium ions start to basically congregate after they've passed into this passed from this part over to here. So they're calling this the inside of the cell here, and this they're calling the outside of the cell. So the potassium ions move through, but then they kind of start to congregate along the membrane. And quite literally what's happening is you're starting to get potassium ions that are congregating over here, they're positively charged, and the anions that can't get through the membrane, they kind of start to get attracted across the membrane to those potassium ions. And so you start to see this charge difference across the membrane, where there's positively charged things right along the inside of the membrane here, right along the outside of the membrane here, and there's negatively charged things over here on the inside of the membrane. So we have, at this point, this is what's referred to as a membrane potential. We have a charge difference across this membrane. And this charge difference is what we're going to be focusing on in this chapter. So whenever you've got the situation where there's sort of an imbalance in the ion flow from one side of the membrane to the next, you'll get these differences where one side of the cell will be a little bit more positive and the other side of the cell will be a little bit more negative. So I have to end there. That's going to be our concluding point. We will pick that up after the quiz on Friday and keep going with it. Yeah.
So you said that